So we're going to start the last part of the program, the second part of the program. If everybody could find their seats, and we could, of course, chat more after this, after the program is totally over. And I will introduce now the Goldman Rakesh Prize. The Goldman Rakesh Prize today goes to Elizabeth Phelps, who I will introduce I will introduce now. There's been amazing congruence of the people who have presented here. Katie has just talked about the amygdala, has just talked about threat, and now we're going to get somebody who's really studied threat as a basic phenomenon and how the brain responds to threat in animals, and then was successful in translating those kinds of threat paradigms to humans. In fact, Elizabeth worked with Joe Ledoux, who actually developed all of the threat circuitry and showed its relationship to anxiety in mice. Of course, mice are not people, so it waits for somebody like Elizabeth to apply it. Elizabeth was trained at Princeton, then she was an assistant professor at Yale, and then she became a full professor at New York University, and then moved to Harvard in 2018, where she is now the Pershing Square Professor of Human Neuroscience. And Elizabeth is going to talk about the human hippocampus. It just seems like this is a continuation of our previous talk. She's going to talk about the human amygdala, threat and anxiety, translational progress, and challenges. Thank you.
So first I want to thank the Foundation for this award. I'm deeply honored to have an award in Pat Goldman Rakesh's name. She was one of my academic heroes. And I want to just talk today a little bit about translation. How is it we take these detailed neurobiological models that we can develop with non-human animals, with using techniques we couldn't possibly use with humans, and translate them to human function? I'm just going to start with a point. So sometimes translation works well, and sometimes translation is imperfect. And so one of the themes of my research has been to say, when is translation working, and when is it not working? So I'm going to talk today about this question: how well do animal models of the neurobiology of threat or anxiety translate to human function? And again, I want to highlight the importance of animal models. I'm a big fan of research with non-human animals. Excuse me. I'm inspired by it. I collaborate with folks who do this research, but I know it has significant limitations, at least in terms of thinking about the complex human experiences we care about. And so I want to talk about translation really on three levels. When we think about a model system in non-human animals, we can ask the question, if we do something very similar in humans and have similar output measures, do we see involvement of the same brain circuits? Sort of a direct translation. But we also might want to ask the question, to what extent do these simple simple behaviors we can study in non-human animals and the brain circuits we identify extend to the complex social and emotional situations that are typical of everyday human life? And then we can ask the third question, which is one that this foundation cares deeply about, which is, how can we use these innovations from animal models to inform new treatments for psychiatric disorders? So I'm going to talk about translation on all three levels. And I'm going to focus on this primary paradigm that's been used in rodent models to study threat processing, which is Pavlovian threat conditioning. This is just a very simple paradigm, and most of the research we know from rodent models about the neurocircuitry that we think is related to anxiety comes from research with this paradigm. And the way this paradigm works is the rodent hears a neutral stimulus, such as a tone. The tone is then paired with an aversive stimulus. After a few pairings, the tone itself comes to elicit a range of responses that we call defensive responses, such as they freeze in the presence of the tone. We see autonomic nervous system arousal, increases in blood pressure, heart rate, and stress hormone release. So using this type of simple paradigm, innovative researchers like Joe Ledoux and Mike Davis, Bruce Kapp, have identified the amygdala as a critical structure in the acquisition, storage, and expression of this simple type of learning. And they've taken that circuitry all the way down to the molecular level, and they've expanded it out to look at brain systems that input to that region and brain systems that control that region. In my work, the very first study I wanted to do in this domain when I was a brand new assistant professor was just ask the question, if we do similar things in humans, do we see similar brain circuits involved? And so we did a very simple study. Take a blue square, pair it with a mild shock to the wrist. Of course, we have lots of ethical constraints in humans, so we had to do this very mild shock. And we measured a very transient, fast indication of autonomic nervous system arousal, sort of changes in the conductivity of your sweat glands and the electrical conductivity from your fingers. So prior to pairing the blue square with a shock, we then saw when participants saw the blue square, they showed a little bit of an increase in the skin conductance response, indicating autonomic arousal. And so this paradigm is a nice translation directly from the animal work to the humans. And what we found, perhaps not surprisingly, is that we see bold activity in the amygdala to the blue square compared to a yellow square that was never paired with shock, along with this increased skin conductance response to the blue square that's absent in patients with amygdala damage. So this just told us this circuitry of this simple learning that you can do in rodents seems to apply to humans as well. But when we think about how we as human beings acquire fears or threat reactions, it doesn't happen all that often that we're actually hurt. If you think about the COVID pandemic, we all took protective action to avoid germs. We've never actually seen that germ with our own eyes. We've seen pictures blown up from microscopes, but we can't see that. We only know about the presence by being instructed about it. This is adaptive for us, right? We don't have to learn by being hurt. We can learn from watching other people, and we can learn by being told about dangers in the environment. And this allows us to adapt in our world to avoid the threats that are out there. But how does the amygdala mediate this type of social learning that really is the main way that people learn about threats in the world? Well, we asked this question in a series of studies. So in one, we had people watch somebody else have a blue square paired with a mild shock to the wrist, and then they were in the same situation themselves. And again, we saw robust activity in the amygdala and this increase in autonomic arousal to the blue square that was paired with the shock for the other person. And we told people, when you see a blue square, you might get a mild shock. And again, patients with amygdala damage don't show this physiological stress reaction, and healthy control participants do, and we see robust activity in the amygdala. So this is important because it tells us that the types of fears that humans experience that may be imagined but never actually experienced engage the same circuitry that we can study in detail in animal models. And this is, again, as I mentioned previously, this is adaptive for us to learn this way, but it also could be maladaptive. We can be afraid of things that will never, ever actually happen. And some individuals can be so afraid of germs, for instance, that they never see that they wash their hands until they're raw. So this could also be a contributing factor to things like anxiety disorders. And this, of course, then begs the question, how do we control these threat reactions when they become maladaptive? And so this is a topic that's also been studied in animal models and in humans. And there's two techniques we have laboratory analogs for that form the basis for the current best evidence-based treatment, sort of psychological treatment, cognitive behavioral therapy. And those two techniques are extinction, and this forms the basis for exposure therapy of cognitive behavioral therapy, and cognitive emotion regulation, which forms the basis for cognitive therapy. So what do we know about animal models, about these mechanisms for threat control, and how do they extend to techniques we can use to control emotion that are more typical for humans? So extinction is something we can study in non-human animals, and we can study in humans as well. Extinction is simply, as Pavlov described it, you have a stimulus paired with an aversive event. You then acquire a threat reaction to the stimulus paired with the aversive event. Now you present that same stimulus over and over again, then there's no aversive event. Eventually, the rodent creates a second memory that this thing is now safe. Humans, the same thing. A blue square paired with shock, blue square, no shock. Eventually, this response diminishes. We know quite a bit about the brain mechanisms of extinction from research with non-human animals. So this is just a slide I borrowed from Greg Quirk, who did a lot of this early work in this area. And what we know is the amygdala, as you've heard a lot about already today, is important in the acquisition, storage, and expression of these simple associations. But when you now want to express your extinction memory, you have to inhibit the amygdala and inhibit the expression of that threat memory to allow the expression of this other memory that in some contexts, this thing is safe. This requires the prefrontal cortex. In rodents, we call it the infralimbic cortex. In humans, we call it the ventral medial prefrontal cortex. And also the hippocampus, which you heard about already, tells us, here's a circumstance or a situation where this thing might be threatening. Here's a circumstance or a situation where this thing might be safe, and it helps you modulate the right context to express these two competing memories. So the first question we asked was, how well does this system that we can observe in rodents, we know quite a bit about, extend to humans? We did an imaging study showing an activity pattern very consistent with what you'd see in electrophysiology in rodents. As extinction progresses, we see a decrease in activity in the amygdala and an increase in activity in this region of the ventral medial prefrontal cortex. In another study, we showed that patients with damage to the hippocampal fail to show this contextual modulation of the threat response. So this tells us, when we do these very similar paradigms in humans and rodents, we engage a similar neurocircuitry. But what about this circumstance? This is not something you can teach a rodent to do. I can't teach a rodent to think about things differently, to see the glass as half full, to imagine that this thing isn't as dangerous as you initially thought and reason why that might be the case. And this is what happens in cognitive emotion regulation, or cognitive therapy, right? You're told to think about, to interpret things differently, and this will change your emotional response. So when we looked in the brain to say what happens when you use your thoughts to change your emotional response, we saw two regions emerge. The same ventral medial prefrontal cortex region that we saw in extinction learning, that we know can inhibit the amygdala, and an additional region, this dorsolateral prefrontal cortex region. This is a region studied by Pat Goldman-Rikish that was involved in the maintenance of manipulation of information on your mind. So we think this is involved in using your thoughts to react to, actively try to change your interpretation to help control your emotions. And this tells us that a circuitry like this exists. We have the amygdala that is important in the acquisition, storage, and expression of these simple associations. The prefrontal cortex, the ventral medial prefrontal cortex, inhibits the amygdala to allow you to express an extinction memory, or the dorsolateral prefrontal cortex takes advantage of this circuitry to allow you to use your thoughts to control your emotional reactions. And this, again, suggests good translation from the circuitry we study in a simple paradigm in rodents to the types of emotional control techniques we can use in humans. So these are great techniques. They are effective clinically, but they aren't perfect. And one of the reasons why we have difficulty with these techniques, why they don't always work as well as we want, is because what we've really done is created a few different representations. One that this thing is dangerous, one that this thing is safe. And to reduce sort of maladaptive threat responses, one has to inhibit the other. Because threat memories and things that are threatening often lead to stronger memories than things that are safe, that often fails, that inhibition fails. And so what you have is poor extinction retrieval in the case of extinction. And there's a number of circumstances that are linked to this. Being in a new context, the passage of time, just psychopathology in general, things like PTSD lead to problems with that circuitry, that inhibitory circuitry. And even just having mild stress can impair prefrontal cortex function, making it harder now to engage these strategies to control your emotional responses. What we really want to know is how can we improve? Oh, and I should say this is thought to be reflected clinically in the relapse rate following cognitive exposure therapy, which can be close to 50%. And so the question we really want to know is, how can we improve this type of intervention to get good extinction retrieval? Can we do something to actually make these techniques more effective? And there's several studies and techniques that have been looked at that are trying to improve this, both in rodents and in humans. I'm going to just highlight one that we've done, looking at this question of, can we more persistently control maladaptive threat and fear responses, perhaps by intervening in the memory itself. So in other words, instead of having two competing memories, one with threat, one with safe, we now change the threat memory itself. To do this, we are taking advantage of a process called reconsolidation. So just so you know what this is, let me walk you through how this works. Until about 20 years ago, this is how we thought memory worked. You learn something, there's a period of time where the synaptic changes that become that memory occur. We call that consolidation. If I did something to block those synaptic changes, like inject a drug that inhibited protein synthesis, which is needed for synaptic plasticity, and I did that right after you learned something, you'd never learn something. It wouldn't get in your brain. It was traditionally thought, however, when you learn something and that memory is consolidated, now that's like a file in your brain. You retrieve that memory, you pull that file out, then you put it back. You retrieve it again, you pull that file out, you put it back. So that memory, once it was there, was thought to be there. About 20 years ago, there was a seminal study that I'll talk about in a second that reinvigorated interest in this other type of technique of thinking about how memory might work. In the reconsolidation view, you learn something, you have that consolidation process, your memory is stored in your mind, but now, some of the times, when you retrieve that memory, a second consolidation process occurs. The second consolidation process also requires synaptic plasticity, and what this suggests is maybe we can intervene with even an older memory during the second consolidation process and change the memory itself. And that's exactly what Karim Nader showed in this seminal paper in 2000 in rodents. He brought rodents into the laboratory. He played a tone, paired it with a shock, waited for that memory to consolidate, then came back. There was a single isolated presentation of that tone out of context, and that's important. And during this second consolidation process, he injected into the amygdala a drug that blocks protein synthesis, which would stop the synaptic changes that become that memory. He came back the next day. He gave the rodent the tone, and it looked as if the rat had never had the association. It looked as if the memory was gone. This was very exciting. This got a lot of attention in the community of investigators who are trying to improve treatments for anxiety disorders. There's one big problem, however. Can't do this in people. It's not safe. So we had to think about an alternative approach. And it's been argued that there are two functions of reconsolidation. One of them is to strengthen memories. You store things over and over, the memory becomes stronger. The second one is to update memories. So when you retrieve a memory, there might be something in the context that's relevant for the future use of that memory. And you can now store that information along with the original memory. Think of it as taking a file out, changing the contents a little bit, updating the contents, putting it back. That's what we think is happening during reconsolidation. So we asked the question, can we take advantage of this updating function of reconsolidation to insert safe information into the original memory during this window of time when we think the memory is essentially being rewritten? So we did that by doing extinction learning after a trigger that would start the reconsolidation process. This was also done in rodents by Maureen Monfils, in my lab by Daniela Schiller, in humans. And what we found was something like this. So here, what I'm plotting to you are two stimuli that were paired with shock during learning. And here's the skin conductance response. This is the stimulus that was never paired with shock. It's the baseline stimulus. So we saw an increased autonomic arousal to these stimuli. Excuse me. The reconsolidation stimulus, we then triggered the reconsolidation process. They did the safety learning, and at the end, in all cases, extinction learning reduced this threat reaction. A day later, however, we brought them back to the context. They got some shocks to say that maybe this is not a safe environment. The group that got standard extinction showed this relapse. They showed a strong threat reaction. Whereas the group that got the safety training during this window of time when we thought the memory was plastic did not show this relapse. It seems as if the memory was in some ways rewritten. We then asked the question, if we look at the brain, do we see these inhibitory mechanisms that we know are important in extinction? And the answer is no. So here is the brain region, the ventral medial prefrontal cortex. We see this increase in the ventral medial prefrontal cortex when people are undergoing extinction, when they're extinguishing their response. So we think this is inhibiting the amygdala to allow the expression of the extinction memory. We fail to see any change in this, for this stimulus that we think underwent the training during this window of vulnerability. So this was very exciting. The original study was published in 2010, the imaging paper in 2013. Lots of people asked the question, maybe we can adapt exposure therapy by creating this window of vulnerability, this reconsolidation window, and then before we do exposure therapy. And would this lead to a greater benefit of exposure therapy? So many people have tried to do that, to translate this to the clinic, to an innovative treatment. And I'm going to tell you the bad part. As of yet, there's limited evidence that this is successful in treating the clinical symptoms of anxiety-related disorders. But I want to walk you through one study that tried to do this to show you why I think this is the case. So this is a study done by Barbara Rothbaum's group, who has been funded by BBRF. And they were trying to use these techniques that we developed to target the fear of flying in people who had a phobia of flying. And they used a virtual reality exposure technique. So people who had a fear of flying came into the clinic. They were asked to go into this virtual reality system where they tried to mimic flying to give them exposure to this so they could learn it was safe. And their only manipulation was, for some of these people, they did this after they created this vulnerability in the memory, this reconsolidation window. For others, they did not. And this is what they found. So this is their skin conductance response. This is the physiological response in the patient to seeing the VR, the VR airplane relative to baseline. And here is the group that underwent standard exposure. Here's the group that underwent reconsolidation updating. As you can see, 3 months later, we have relapse in the group that underwent standard exposure, but not in the group that underwent reconsolidation updating. So this looks like a success, right? They had the same measurement as we had in the laboratory, and they found the same results, but here in a clinical population with looking at skin conductance. The problem was, however, they didn't see any benefit on subjective symptoms. So if you were to give these patients a scale to ask them about their fear of flying, they're going to look exactly the same, regardless of whether they got this treatment. So using the techniques that we adapted from the outcome measures that we adapted from the rodents to the humans, we're finding great translation. Using that same measurement, we're finding good translation to clinical treatment, but not using the kinds of things we normally care about. Is this person reporting distress? Do they say they're afraid? All these subjective things that is the quality that we think about when we think about these mental disorders. So this quite begs the question, how are we doing so far? So what I've shown you is through a series of studies, many more than I'm presenting today, we're finding very good translation when we take these simple animal models and we do very similar things, and in humans, we see the brain seems to operate just the same. So we can have a lot of assurance that all the details of that neurobiological model that we're studying, we can enact the same processes in humans. We see pretty good translation even when we go to these complicated situations that are typical of our everyday life that characterize human fears that aren't relevant to fears in non-human animals. But we're not seeing such great translation when we're getting to the subjective clinical symptoms that we really care about, the kinds of things you can never measure in a rodent. And so this begs the question, how might we do better? And so this is what we're trying to do now. I'm not going to give you any data. It's a little teaser. But the way we've thought about translation in the past is going from the laboratory to the clinic, going from the animal model, and then trying to think, how can I look at this marker in humans, and will it work in the treatment of disorders? But we're finding that's not working so well, particularly when it comes to all of these subjective symptoms that really are the characteristics that we talk about with these psychological disorders. What we're trying to do now is maybe do a sort of circle translational approach, an alternative translational approach. Can we think about the main symptoms we care about and combine them with insights on the neurobiology of threat from animal models, but use them in novel ways to target not just the measures that are similar, like physiological stress, but also the measures that are not similar, like subjective stress. And so those are the things we're trying to do now in my lab, and I'll leave that as a teaser for you. I just want to end by highlighting, which other people have talked about, science is a team sport. This is just a small part of my team, the ones who contributed to the science you saw today, but I couldn't do anything without all of my collaborators and colleagues and those who have funded my work. And I want to thank BBRF for this honor, and also for their path-leading research, or goals to enhance research in this area, and the contributions they've made to research on psychopathology, and all of you for your attention. Thank you.
So, a great talk, but you have now a disconnect between a biological measure.
Excuse me?
You have a disconnect between a biological measure, right, and the symptom improvement.
Yep.
Now the question is there a lag that you might see the improvement over time with repeated, and that you have really now the established synapses and everything else in the circuitry that it will take time to dissolve the incorrect associations?
That is possible. Certainly possible that there's going to be a feedback, right? Once you sort of reduce some of the physiological responses, you then start to reduce some of the other more subjective responses. But there's a lot of evidence, and there's some seminal research on this that everyone seems to have forgotten, like it's about in the 70s and even the 60s, showing a lot of disconnect between the physiological symptoms and the subjective symptoms in things like anxiety. And so, there probably is a feedback relationship, but it's certainly not a perfect relationship. We know, for instance, the amygdala, that we've talked a lot about, is a brain region, that is talked about as being an important emotion, but most patients with amygdala damage report normal subjective threat and fear responses, right? There are rare examples of cases where they don't, but the vast majority of them do, right? So it's probably, a different representation that certainly feeds back with the physiological representation, but I think you're going to have to target both if you're really going to make progress. And we need to understand more about the representation in the brain of those subjective symptoms as well.
I'll slip in a question. Do you think that humans, excuse me, Elizabeth, I'll just slip in a question.
Sure.
Humans have more to process, so we need to be a little bit broader about what we're thinking for them? Or do you think that we just have to keep plowing through the possibilities?
I think, because we've been so inspired by the animal models, we've ignored many of the things that we as humans think about, right? So is this broader than, I can't get in the mind of a rodent, right? But we have a much more complicated set of cognitions, a much more complicated way of learning about threats, and we have all these subjective qualities, in consciousness and things that we can, that we can reflect on. So I think the rodent work is a good place to start. But because we've been so inspired by the rodent work, I think we've ignored these subjective measures. And I'll just make the point, Joe Ledoux, he was talked about in the introduction, and also we collaborated for a good 25 years, longer than I want to admit. He recently, he used to call this fear conditioning, and he recently decided that he shouldn't be using the word fear. He calls it threat conditioning, and these defensive responses, because fear is something, that we experience. If I say fear, you know what that is, right? It's clear the rodent is threatened. We don't know if the rodent has a fear feeling, right? So Joe has sort of been a big advocate in the last decade to saying, when we're talking about rodent work, let's talk about what we're measuring and not assume a cognitive state that's unique to humans.
You were fantastic, by the way.
Oh, thank you.
Yes. Yeah, my name is Chiffon, and I just wanted to ask you about the reconsolidation part that you so eloquently shared. So here's what I'm trying to understand.
Yep.
If there is a reconsolidation that can happen for someone who's experiencing mental health illness, let's say, is it safe to assume that if someone has post-traumatic stress disorder, let's just say, are you saying that there's a possibility that they can be trained into rewriting the communication that happens inside certain areas of the brain to alleviate that disorder or illness?
Well, let me say, Siobhan, I think that's the hope of any type of psychopathology treatment, right? That we can sort of rewire the brain to make it more effective. The reconsolidation work is really promising because it suggests perhaps particular time windows that maybe we can identify that might be more effective for some of those treatments to work relative to other time windows. I will say the big challenge in reconsolidation, and some of the work we're doing now, is that not every time when you come up with a memory does it undergo this second kind of plasticity, only sometimes, right? And we're trying to understand why that's the case, and can I look at the brain and know that this is a memory that's going to be something that I might be able to modify or change versus one that's not. And the other point I'll make about reconsolidation, my colleague Christina Alberini, again, argues there's two functions. One is strengthening memory, right? And she thinks that's the primary function of reconsolidation. The other one's updating memory. We know memories are dynamic. We know that memories change over time. This is just a mechanism by which that can occur. But it also can strengthen memory. So, these multiple retrievals that you have with no sort of intervening information could actually just be making the memory stronger and stronger and stronger. And I think we also have to balance sort of those two possible outcomes. That we're not strengthening the memory by doing this. And in fact, I can do studies of just having people recall words, and if I just remind them of the context, I can strengthen that memory. And so it's very complicated. It's a process, I want to point out. It's a natural process. This is happening every day as we speak, right? But we just are now understanding the timing of it and how it works, which may allow us sort of a new window of therapeutic intervention, but we don't know yet. It hasn't gotten there yet.
I have a cousin who has been terrified of dogs forever, and has been in therapy. I wonder if the dog is really not the problem. I mean, maybe it's something else. Maybe the dog stands or something. I mean, what do you think?
Yeah, I mean, it's possible. It's hard for me to say. I don't know.
It's possible.
Yeah, I don't really know. I will say about, a lot of phobias, this gets to the point of things being imagined and experienced, but not ever really experienced. I mean, the idea of phobias, emerging from a bad experience, often people don't identify a bad experience, right? And the bad experience can simply be something that you imagine from watching somebody else or being told by somebody else, right? And I think that what's important about the first series I talked about, that tells us that same brain circuitry, it's like the fear circuitry is still the fear circuitry. It doesn't matter if it's in your head or it actually happened to you. Yep.
Thank you.
Thank you.
Following your work for years.
Oh, thank you.
And a fan of Joe Ledoux, and his change to threat makes
No, I think, Joe,
huge difference.
Joe has always been a big advocate of let's be really precise about what we're talking about as scientists. So.
I have a question. What you are describing, does that become part of the default mode network? And if you then
do the changes you're describing, would it change the default mode network?
I don't, it's not, the default mode network is bigger than just the brain circuits I'm talking about. So I don't know if anyone's looked at that, but I don't think it would change the default mode network. That's just my hunch. So,
Do you think
Is,
I don't know if we want to.
thank you.
Hi.
Is there a tendency of people with mental illness to create false memories because of the earlier discussion of people with schizophrenia where their self-assessment is poor, and therefore then when they are speaking of fearfulness, anxiety and stuff, and even just, feeling lousy and things, it's of things that really never happened?
So, I mean, I think you saw some evidence earlier that, yes, people, with mental illness, at least some can create false memories. But, I kind of don't like the word false memory, and let me tell you why. We have memories for things that happened in the world, and we have memories for our thoughts, right? And so when we call them false memories, they're really just misidentifying the source. Was it something I thought, or is it something that actually happened to me? In brain studies on false memories, brain imaging studies on false memories, there's very few distinctions of those things in the brain. You can find a little bit of activation in perceptual areas that distinguish them, but yeah. Yeah. Yeah, I mean, so I haven't studied that, but I do think, to the extent that all of us engage in these processes, as you interpret information, it's going to also get incorporated into your memory. So if they're interpreting information in the world differently, that will also be part of their memories. So,
Elizabeth, thank you very
Thank
much
you.
for your great talk.